Takže dobrý den všem, já jsem Michaela Oborská a především chci poděkovat organizátorům za konání téhle premiéry, tohle meetingu. A předem se omlouvám, mám teda připravenou prezentaci v angličtině a předem se omlouvám, že je to teda hodně vědecký. Full screen. Takže když neoslovím všechny vás, tak se předem omlouvám. So, hello everyone, I am Michaela Oborská and I came roughly 5 years ago to Zurich to start my PhD in the scientific group of Vikram Panse. Back then at Institute of Biochemistry at ETH Zurich, but now we are located in Institute of Medical Microbiology at university. And I successfully finished my PhD last week, so we should later cheer on it together. And during my PhD, I studied how the large ribosomal subunit is actually crafted in a cell, how it's assembled. So, as the title say, in Vikram's lab, we study how the functional ribosome in the cell is assembled, and which is a fundamental structure which is responsible for the synthesis of the whole proteome of the cell. An eukaryotic ribosome composes of two ribosomal subunits, large one, or 60S, and the small one, 40S. And each of these ribosomal subunits composes of ribosomal RNA, here depicted in gray, and ribosomal proteins, here in color. And the assembly of this structure in the cell is a very complex process, which spreads through distinct cellular compartments in the nucleolus, nucleoplasm, and the cytoplasm. And the whole process starts in the transcription of the primary transcript, the RNA, in the nucleolus. And then transcriptionally, ribosomal proteins, which are synthesized in the cytoplasm, are transported to the nucleus to co-transcriptionally assemble with the ribosomal RNA. And there are also more than 200 assembly factors, which are not part of the mature structure of the ribosome, but which are there to regulate the whole pathway and steer the pathway from the nucleolus to the cytoplasm. And they co-transcriptionally join with the ribosomal RNA to form the earliest precursors of the ribosomal subunits, whose maturation pathway is separated very early on by co-transcriptional cleavage of the ribosomal RNA. The maturation pathway progresses through the nucleoplasm as a hierarchical cascade of events, which eventually renders the ribosomal subunits export competent. And after the export of these particles from the nucleolus, they undergo the last maturation steps and quality control checkpoints before entering the translation. And the ribosome biogenesis, the whole process, and especially the early steps, involves a robust RNA folding. And you can imagine it as like a string of RNA which needs to reach the functional fold. But this occurs via transiently misfolded trapped intermediates. And to reach the functional fold, cell is equipped by RNA proteins and RNA chaperones to steer to reach this functional fold. And now the low temperature is actually a kind of a barrier for RNA folding, which accumulates and favors these kinetically trapped intermediates. So we were wondering how yeast cell, would react, would respond to exposure to low temperatures, which is this unfavoring condition which kinetically traps these misfolded intermediates. And to do so, we exposed yeast culture to distinct temperatures and quantified the abundances of the proteins in dependence on the temperature by quantitative mass spectrometry. And then we gathered kind of this cluster of behavior of these peptides in dependence on the temperature. And we got 9 distinct clusters of this behavior, how proteins are behaving, their expression independence of temperature. And we were mainly interested in these clusters showing the highest abundance of proteins at low temperature, 20 degrees, with decreasing trends in increasing temperature. And we found that these clusters actually enriches ribosome assembly factors and RNA binding proteins. So these factors are basically globally upregulated at low temperature in the yeast cell to assist the RNA folding in this unfavoring low temperature condition. And we further dissected the role of one of these low temperature induced assembly factors, which is called Puf6. Puf6 has an RNA binding domain, which looks like this helical repeat kind of domain. And you can see that we just confirmed that Puf6 expression is boosted at this low temperature. Now, these factors, when we delete it from the cell, it's not essential, but you can see that we see a slow growth phenotype at low temperatures, which nicely correlates with this higher expression profile. Next, we wondered whether Puf6 is involved in the ribosomal pathway or maturation pathway of the large or small ribosomal subunit. We do that by GFP reporters. So the GFP is fused to either ribosomal protein of a small ribosomal subunit or large ribosomal subunit. And then if there is no ribosome assembly defect, we would see these reporters in the cytoplasm. But if there is some defect of the ribosome assembly, we would see nuclear accumulation. And this is exactly what we see for Puf6 delta cells, specifically at low temperature, where the phenotype also is, the growth phenotype, and only the nuclear accumulation we observe for the large ribosomal subunit, not for the small ribosomal subunit, meaning that Puf6 actually works in the maturation pathway of the large ribosomal subunit. Now we wanted to dissect at which stage of this maturation process Puf6 acts, and we can biochemically purify the stages of ribosome pathway. And then we could detect Puf6 associated with the earliest stages of ribosome biogenesis, showing that Puf6 acts early on in these early stages of the large maturation pathway. And the last puzzle we missed was where exactly Puf6 binds on the large ribosomal subunit, and we used it by a method which is based on the UV. We cross-link protein to the RNA, and then we detect this RNA piece where Puf6 binds. And we detect this binding site in the helix 60, predominantly in helix 68, which maps on the subunit interface of the large ribosomal subunit, where the all functional centers actually lies. And we further dissected which piece, we narrowed down the binding site with another method, which study RNA-protein interactions. And we take advantage that if there is an interaction between the RNA and protein, the reporter gene, in this case histidine, is expressed, and the cells are able to grow on plates lacking histidine. So we confirmed our previous experiment with this method, showing that the yeast helix 68 can bind, growing on the plates minus histidine, Puf6. But equivalent helix from a bacteria doesn't bind Puf6. And then we took advantage to kind of define which piece of the yeast helix 68 binds Puf6 by just creating a chimeric constructs. And we could see that if we have the tip of the yeast helix 68, we got binding. So the Puf6 binds to this tip region of helix 68 from yeast. And this tip of the helix 68 on a mature ribosome is involved in this peculiar tertiary contact on the mature ribosome between helix 22 and 88. And to put it into the context of the ribosome, here's the secondary structure of the large subunit. And it creates kind of 6 independent domains, which needs to come together and compact into this large ribosomal subunit. And this tertiary contact kind of joins a distant part of this subunit. So it compacts the whole ribosomal subunit. And we were wondering how Puf6 actually, how acts on this. What's the impact or function of Puf6 in this tertiary contact? So we reconstituted this tertiary contact in vitro, just taking the single helices of this tertiary contact, joining them with flexible linkers. And we studied the folding of this construct in vitro. And the tertiary contact or RNA folding can be promoted by addition of monovalent and divalent ions. And in this system, we saw that the monovalent ions, such as potassium, promoted formation of this kissing loop between this helix 22 and 88. And then magnesium promoted the docking of the helix 68 into this tertiary contact. And then when we add Puf6, our player, into this role, we saw that Puf6 actually acts the same way as magnesium, meaning that Puf6 compacts this tertiary contact and form this tertiary contact, chaperone it. And when we look at the available structures of ribosome intermediates from different states, we could follow kind of a formation of this tertiary contact on the pre-ribosomal particle. These are particles from the nucleolus, very early stages, and we could see that initially the helix 22 is shielded by another assembly factors, BRIX1 and EBP2. When these two factors are released, this helix is positioned in a way that it can engage with this helix 88 to form this kissing loop. And then later on in the nucleoplasmic particles, we could see that there is already this tertiary contact formed. So we assume, based on our in vitro data, that our player would have to act in between these two stages to dock helix 68 to the kissing loop. And to test, so we would assume that if Puf6 is not here, this wouldn't occur and have consequences later on the maturation pathway. And later on, in the pre-ribosome, helix 68, which is docked and formed this tertiary contact, is binding site for another assembly factors called SDA1 and NOC2. And NOC2 is an important player as a checkpoint protein before allowing the nuclear export of the particle from the nucleus. And we hypothesize that if this helix 68 is not docked in this position, this binding site couldn't be properly formed for these two factors, and their association with this particle would be impaired. So we approached this biochemically, and we purified these pre-ribosomes from wild type and Puf6 delta cells at 20 degrees, and we could indeed check that the association of these two assembly factors is impaired, and so that Puf6 therefore primes an 60th pre-ribosome nuclear export because of this formation of tertiary contact here. So to summarize it, this is like a final model. So the large ribosomal subunit the RNA is co-transcriptionally made and fold in a hierarchical manner. And now Puf6 early on associate with the ribosomal RNA via helix 68, which is located in the domain 4, and awaits for the formation of the kissing loop between helix 22 and 88, and only after that, Puf6 docks this helix 68 to form this tertiary contact, which compacts the whole particle and enables the progression of the downstream steps, such as efficient nuclear export. Yeah, and then with that, I would like to thank all my group, mainly my supervisor, and all past and current members of my group, mainly Stefan, who initiated this project and let me finish it. Of course, my thesis committee for nice discussions during thesis committee meetings, and mainly also to collaborators, Richard Werner from Siegel Group, who helped me with the reconstitution of the in vitro RNA folding assays, Ludovic, who actually helped us with all the quantitative mass spectrometry, and Rob, who helped us to crack the binding site of the Puf6. Then also the technical stuff from both of my institutes and founding, and here I would like to highlight Beringer Ingelheim Fonds, that I got a fellowship from them to pursue this project. And thank you for your attention and coming.
Tak děkujeme za úžasnou prezentaci a máme prostor na jeden dotaz. Z časových důvodů. Tady máme.
Děkuji. Slyšíme se? Jo, dobrý. Děkuji moc za prezentaci, byla zajímavá. Většině jsem asi nerozuměla úplně, ale co by mě zajímalo, vy jste teda zkoumala něco, kvůli čemu jste to začala zkoumat, nebo jakoby k čemu se to dá použít, tak jaký to má využití?
Tak ribosomální biogeneze je základní proces, je to fundamentální proces a vědět detaily z toho procesu, který potom můžeme využít k tomu třeba navrhnout antibiotika, který třeba by targetovali ten helix, aby zastavili třeba formaci z toho celého ribozomu. To jsou potom samozřejmě možná trošku science fiction v této podobě, ale myslím si, že je důležitý rozumět veškerým tím detailům těch procesů, zvlášť když to ovlivňuje složení něčeho, co můžeme potom využít jako potenciální drug target, třeba v rakovině, v rakovinných buňkách, anebo právě i vlastně efektovat, já nevím, antifungální třeba, teda navrhovat antifungální drogy, drugs, léčivo. Drogy.
Drogy. Tak, děkujeme moc ještě jednou.